In this video, I'm going to go over the stages of mitosis. Before we get into mitosis, though, I just want to kind of recap what we're talking about here. So we're looking at the somatic cell cycle. So this simulated cell I have on this whiteboard would be something like a skin cell or a muscle cell, any cell other than the sex cells. Your somatic cells are diploid, meaning that they contain two sets of DNA. They would have your maternal DNA, so DNA from your mom, and your paternal DNA from your dad. And those come in 23 pairs of chromosomes for a total of 46. So we can just show maybe the maternal DNA here. And I'm only going to be dealing with one chromosome. We're going to be dealing with chromosome 1. And then the paternal DNA here. Okay. And this is showing a cell that would be in, say, interphase, whether it's G1, S, or G2. Now, in S phase, if you remember, before the cell divides, so this is all before we get into mitosis, before the cell divides in S phase, the DNA is going to all replicate. So we're going to see a replication of all of this DNA. And that's an important step, because if we're about to make two new cells, then both cells are going to need a set of DNA. So that's an important idea. So whenever we get into prophase, which is going to be the first step in mitosis, so this is assuming that interphase has concluded. So this is in prophase. The chromatin is going to wrap up into chromosomes. That's the first step. So we're going to see all of this chromatin wrap up into chromosomes. And I'm going to show those chromosomes as just these little pieces of these popsicle sticks. So we wouldn't only have one copy of both the dad's chromosome 1 and mom's chromosome 1. But since the DNA replicated, we actually have two copies. So those will match up next to each other here. So we have two copies of mom's chromosome 1 and dad's chromosome 1 here. We would call those homologous chromosomes. They're homologous chromosomes because they are the two copies of the same chromosome, one from mom, one from dad. The only reason that there's two of them is because this DNA has been replicated. So they are homologous chromosomes. But what we're also going to see here is that the two replicated copies are also going to get a name. And those are called sister chromatids. So, chromosome 1 from dad, chromosome 1 from mom. Those are homologous chromosomes. The two copies of dad's chromosome 1 are sister chromatids. Also, the two copies of mom's chromosome 1 are called sister chromatids. So those are some important vocab words to keep straight. Move those out of the way. And we'll continue on our journey here in mitosis. Okay. So we now have these two copies of both mom and dad's chromosome 1. And you would imagine that normally we would see all 46 chromosomes existing in pairs right now for a total of 92. I don't have room for that, so I'm only going to show one chromosome. So chromosome 1 and 2 from both mom and dad. So in prophase, we see a few things happening. First is that chromatin condensing into chromosomes, which I've already shown. The other is this organelle called centrioles, which I'm going to show by drawing a box with an X through it. That is not the actual structure. I'm just trying to provide some kind of representation. And these are called centrioles. They're going to migrate to opposite ends of the cell. They don't normally exist at opposite poles like this, but during prophase, they're going to migrate to opposite ends of the cell. So those are these centrioles. Now what they produce is this special fiber called a spindle fiber. I'll use blue for one side and purple for the other so we can keep these squared away. These are called spindle fibers. And I like to think of spindle fibers as being kind of like ropes. Their goal is going to be to latch on to the chromosomes so that we can start moving these chromosomes around. It's going to be important that we have a way of moving these because we're going to have to get each set of DNA, so both a copy of mom and dad's chromosomes, to opposite ends of the cell, so we can end up splitting this cell in half and making this one cell into two. So the spindle fibers are actually going to come down, still in prophase, and attach one to each sister chromatid. And we'll see the same thing on this other side. And that's going to conclude prophase. So in prophase, just to recap, we're going to see the chromatin condensing into chromosomes, the centrioles migrating to opposite ends of the cell, and then the spindle fibers coming out of the centrioles and attaching to the sister chromatids. So these spindle fibers from one side of the cell will attach to both one copy of the maternal and paternal chromosomes, and the same thing will happen on the other side. And that ends prophase. So reset this real quick. And we need to move to metaphase. Okay. In metaphase, these spindle fibers are going to start pulling on the chromosomes. And in order to keep everything organized, because remember, we're going to be dealing with 92 chromosomes, not just 4. In order to keep everything organized, the spindle fibers are going to be moving these chromosomes straight down the center of the cell. And you could imagine having all 92 chromosomes stretched across the center of the cell. And the spindle fibers are still attached. Notice that one spindle fiber from each side is connected to both a maternal and paternal chromosome. The location that these chromosomes are organized on, straight down the center of the cell here, right down the equator, is called the metaphase plate. And the metaphase plate, I have these flipped. We're in metaphase. The metaphase plate is this location straight down the center of the cell. So I'm going to show that with a dashed line. That is the metaphase plate. It's not an actual physical location. It is just an imaginary line, kind of like the 50-yard line on a football field, that shows the location that the chromosomes are going to align down. So it's not an actual structure within itself. It receives its definition from the chromosomes being there. So in metaphase, this is pretty much all that happens. Since the spindle fiber is attached to the chromosomes, it's able to actually move the chromosomes around and get them into this central location down the metaphase plate. This creates a highly ordered scenario where the chromosomes now are going to be able to be pulled in opposite directions and ensure that all of the DNA that was replicated ends up on both sides of this cell. Remember that in mitosis, the goal is to replicate the nucleus. So we're attempting to replicate the nucleus of this original cell right now. All right, so what we're going to see in the next phase now in anaphase is we're going to start seeing these spindle fibers pulling in opposite directions, which is going to allow these chromosomes to move towards those two poles of the cell. So that concludes metaphase. That brings us then to anaphase. In anaphase, where these chromosomes are attached by the spindle fibers, they're going to be pulled in opposite location, or in opposite directions towards the two poles of the cell. This is kind of like an action shot whenever you're in anaphase. So we're actually catching these chromosomes moving during anaphase. So really they'd be a little bit further up. So in anaphase, the sister chromatids get